Hello students, welcome back to botany class. Today we are seeing the problems and molecular basis of inheritance. That is also a very simple problem. Now we will start, then we will go with the another kind of the problem. So the problem is in a double stranded DNA molecule, see the problem. In a double-stranded DNA molecule, both the strands were labeled with heavy isotopes of N15 and were transferred to the N14 medium. So, it undergoes replication for 3 generations. What is the ratio of DNA molecule with both the strands with, that is N14 N14 and N14 and N15 hybrids? So here we are seeing this type of problems which are going to be done only in the bacteria. So for the binary fission of the bacteria, it takes only 20 minutes for doing the binary fission. So here we are saying the same thing. Okay, so what we are saying here, the 3 generations, they asked the 3 generations takes 60 minutes time. So they have given the 3 generations. That means in the E. coli organism, binary fission occurs in every 20 minutes. That is the 60 minutes time. Okay, so in this one, 20 minutes, those 3 generations we can say. So here, first of all, we have taken the organism DNA completely with the N15, N15. Now this was grown on N14 medium. So here the color indication is very clear. Green color giving the indication of the N15 strands, and the orange color is giving the indication for the N14 strands. Okay. So after the first generation, when we have grown, then it will be taking up the N14 to make its genetic material. So we are having the 2 offsprings with the N15, N14, N15, N14. Okay, that means 2 hybrids we got it. So again the second generation, that is the again the 20 minutes now. So from this organism we are getting 2 offsprings, and from this organism we are getting 2 offsprings. That means total 4 offsprings we can expect from this. So this is the first one, second one, third one, fourth one. So here one strand 15 separates, 14 separates. So this 15 will make a new strand with the culture medium, that is of the 14, and here already one 14 is there from the parent. So one more new 14 was made, N14 strand. Okay, like that here also. So in this 4 offsprings, 2 are hybrids, 2 are normal, that is 50-50. Okay, here all are hybrids, here 50-50. That means 2 offsprings with the hybrid varieties and 2 offsprings with the N14, N14 strands. Now we have taken the third generation. In the third generation, we will get total 8 offsprings. See here, 1, 2, 3, 4, 5, 6, 7, 8 offsprings. In these 8 offsprings, 2 offsprings are having N15 and N14 strands in them, and all others will have the N14, N14, N14. So with this, what we can get out of 8, 2 will be the N14, N15 hybrid varieties, and the all others, that means total, in the total 8, 2 are the hybrids. That means 8 minus 2 we are getting as the 6 are the remainings. 6 remain will be having the only the N14 strands. So here 6 are homozygous, that is the N14, N14 strands will be there. So here they are asking us the ratio of the DNA molecules. Then we have to come to the 6:2. What he said again here, N14 to N14 and N14 and N15. That means 2 strands should be normal and 14-14, and the another will be the hybrid variety. So like that we are getting the 6 offsprings with the normal N14 N14 strands, and 2 will be the N15 N14 strand. That is, if we simplify this number, we will get the 3:1 ratio. Okay, like that we have to give the answer. And the same problem they can ask in another way. How they can ask? Instead of here 3 generations, they may say it as the completely 80 minutes. That means in a DNA molecule, both the strands were labeled with heavy isotope N15 and it transferred into the N14 medium. Then it undergoes a replication for 80 minutes. Okay, now here they will say not the 4 generations. Same type of problem, but the case will be the different one. How they will ask is the different. So here they may ask, apart from this 3 generations, they may ask with the 80 minutes. Then what happens? See here, till here the question is same. Then it undergoes replication in 80 minutes. Then what happens? Then what could be the ratio in this? Okay, so they are asking again the ratio, but in 80 minutes. So if we are taking the replication process occurring from the one organism which is having the N15 N15 strands both, just like in this same one, N15 grown on N14 medium. Now we got N15 N14 N14 strands in the first generation. Like that, if we take the next 3, that is total 16 offsprings we will get over here till 80 minutes. First 20 minutes, the 2 offsprings. Another 20 minutes, we are getting 4 offsprings. And the third generation, that is 60 minutes, we are getting 8 offsprings. In the 80 minutes, that is the 4th generation, we are getting total 16 offsprings. In these 16 offsprings, how many are the hybrids and how many are the normal ones? So what we have to say, so out of 16 offsprings, what we are saying, 2 are always hybrid, that is the N15 and N14 strands, and the rest of them are the N14 strand. That is, in the 16, 2 are the hybrids. If you deduct the 2 from the 16, then we are getting 14 with the N14 strand. That is, both the strands are homozygous. Then in this, the ratio can be N14 to N15. That is 14:2. That is, when we simplify, that is comes as the 7:1. And the same one, if we are taking with another words, same question, but they won't ask us now the ratio. Here what they will ask, the after 4 generations. Okay, otherwise after 4 generations, after 3 generations, what could be the percentage of these organisms? When they are saying the percentage, 3:1, that is 75 and 25%. But see here, in this one, we are not saying completely 75% are of the N14. That means only 2 are the N15 strands and all others are the N14 strands. So in this, it cannot be the 75. More than 75 are the N14. So in the options, blindly what we can take it as 87.5% and the 12.5%. If 50% they have given the option, then that is the wrong one. And if they are saying the 70:30, that is the wrong option. Okay, and 60:40, that is also wrong. More than 75, less than this. So it is coming as 87.52 and the 12.5%. Okay, like this, if the ratios, if the percentages are like this, then we have to opt like this option only, not any other option in this. Okay, so prepare with this type of problems like this only, so that it's easy for us to go for the problematic solvation in the NEET exam as well as in the EAMCET exam. And one more thing will be there. See here. Now the length of bDNA, that is a type of the DNA, is called as the bDNA, okay, which is having 45,000 base pairs. Then how many complete turns will the DNA molecule takes place? That's the question what they will ask us. So whenever they have given the base pairs, so 45,000 base pairs, make it this in the brain. So one turn in the DNA, we know that it is having 10 base pairs in them. Okay, so like that 45,000 base pairs. So what could be the length of the DNA? 45,000 by 10, that is 4,500 complete turns it will take in the with this type of base pairs. It's a simple one. The number should be divided with the 10, then the answer will come. Okay, remember, because in one turn 10 base pairs. Like this, here they have given the base pairs, but the turns in they are asking. Okay, so this one divided by the basic number, that is 10, then we are getting the option answer as the 4,500 complete turns it takes. Okay, and the next one more thing is the how they can ask how many hydrogen bonds formed between two strands of DNA of 170 angstroms length if the cytosine is of 20%. Then the length of the base pairs will be 3.4 angstrom, that was the standard one, okay, fixed one. And DNA segment length will be the, now what they have given, 170 angstroms. That means 170 by 3.4, it becomes the 50 base pairs. Okay, 50 base pairs. And in this one, 20% are the cytosine are there. 20% cytosine means the equal amount should be there with the guanosine, that is the Chargaff's rule. Adenines, and that is the purines is equals to pyrimidines, and purines will always pair up with the pyrimidines. So with this, what we can say, how many cytosines are there? Those many guanosines should be there. Okay, guanines should be there. That means 20% guanines. So total 20 plus 20, 40. Okay, now in this, the rest of the one should be the adenine and the thymine, that is 60%. That means adenine 30, thymine 30. Okay, so this will be making the, what they are asking, the number of formed between the two strands. What is the, if the cytosine is of the number of hydrogen bonds they want. How many hydrogen bonds? Then the always guanine pairs up with cytosine with the triple bond. That means 3 hydrogen bonds. So here total is the 40% now. So in this total number, total base pairs we have to take. Total base pairs we are having 50. 50 into 40 by 100, that is becomes the 20 base pairs. And 20 base pairs will be present in between the guanine and the cytosine. And like that, if we take with the adenine and the thymine, those are of the 60 base pairs. So like that we will get the and here this one, and this is the base pairs. Now hydrogen bonds we want, that means triple bond now 20 into 3 is equals to 60. So like that here also 60. So total 120 hydrogen bonds we will get in the in this answer. Okay, so prepare like this. If you are having any doubts, you can put a query. Okay, you can ask sir, so that we will give you the remedy for the problems. Okay, right, thank you.